Here we have the first lecture in the section on the nervous system. It's called Fundamentals of the Nervous System and Nervous Tissue. This is a block diagram showing the basic components of the nervous system. We'll look at each one of these shortly. First of all, there's the central nervous system. Central nervous system is basically the brain and the spinal cord. It contains all the integrating and control centers of the body. It interprets sensory input and dictates motor output based on the sensory input, on reflexes, on current conditions, and on past experiences. Now, connected to the central nervous system is the peripheral nervous system, and that's divided into typically the cranial nerves and spinal nerves. These peripheral nervous system is divided into sensory and motor divisions. The sensory divisions are afferent fibers, and that brings information up to the central nervous system. The motor division are efferent fibers, which brings information away from the central nervous system and downward to the organs and to the muscles and glands. So afferent nerves bring sensory information to the central nervous system. Efferent nerves bring information away from the nervous system. The motor division is divided into somatic and autonomic nervous system. The somatic nervous system is basically the voluntary skeletal muscle system. These are the motor neurons that control the skeletal muscles. These are all voluntary control and under conscious control. As opposed to autonomic nervous system, which regulates smooth muscle, cardiac muscle, and glands autonomically. In other words, you don't have to think about your heart rate. You don't have to think about the glands secreting. You don't have to think about the digestive tract, peristalsis, and that sort of stuff. It just does it automatically. There's two divisions of the autonomic nervous system. There's sympathetic and parasympathetic. We'll discuss that more in the lecture on autonomic nervous system. But you might recall from the histology, when we looked at the histology of the nervous system, there was neuroglia and neurons. The neurons are this large structure here. This is a neuron cell body. This is probably an axon. These are probably dendrites, the different processes that are affiliated with the neuron. These other cells are neuroglial cells. These are support staff cells. These help support and provide functions to make life better for the neurons themselves. We'll look at some of the neuroglial cells. In the central nervous system, there's 4 neuroglial cells: astrocytes, microglial cells, ependymal cells, and oligodendrocytes. In the central nervous system, in the peripheral nervous system, there's only 2 neuroglial cells: there's satellite cells and Schwann cells. Let's look at these. Astrocyte, or star cells, as the name astrocyte literally means star cells. These are the most abundant neuroglial cells. They cling to neurons, they cling to synaptic endings, they cling to capillaries, and they support neurons, they brace neurons, they help stabilize the connection of neurons, they help young neurons find their way to the synapses that they're supposed to go to. They're basically one of the major supporting cells for the neurons. Microglial cells are sort of the healthcare providers for the neurons. They monitor the health of the neurons and they migrate towards injured neurons. If needed, they can transform into macrophage-like cells to phagocytize microorganisms, bacteria, and pathogens, as well as debris from destructed or injured neurons. Ependymal cells. Inside the brain and the spinal cord, there's cavities or ventricles or areas filled with fluid. These ependymal cells line these fluid-filled areas inside the brain and the spinal cord. They have cilia, these little hair-like structures. They help move the fluid. The fluid is called cerebrospinal fluid. This is the fluid that comes out when someone gets a spinal tap. But these ependymal cells help keep the fluid flowing through the ventricles and through the spinal cord. And the last central nervous system glial cell is oligodendrocyte. This wraps the central nervous system nerve fibers with myelin sheaths. Myelin is a substance that's produced by the oligodendrocytes that increases the speed of nerve conduction. Now, looking at the peripheral nervous system, there's two glial cells, satellite cells and Schwann cells. The satellite cells are basically the same thing as for the peripheral nervous system as astrocytes are for the central nervous system. They have very similar function. And the Schwann cells are very similar to the oligodendrocytes. So the Schwann cells in the peripheral nervous system are similar to oligodendrocytes in the central nervous system. Here's a picture of a neuron with the satellite cells around the cell body of the neuron. Here's a neuron axon, and it has these little areas of Schwann cells that are wrapped around it. This actually helps the nerve conduct electricity or faster than without these myelin sheaths. Now let's look at neurons themselves. The neuron is the structural unit of the nervous system. These are cells that are specialized to conduct impulses. They conduct electrical impulses from one place to another. They have very long longevity. They live the lifespan of the person that they're found in, so they can live 100 years or more. They're amitotic. That means they don't undergo mitosis. They don't have the ability to divide. Once a neuron is made, it doesn't divide and have baby neurons. It stays until it dies. Neurons have a high metabolic rate. They require a lot of oxygen and a lot of glucose to function. They all have cell body and one or more processes. The processes could be dendrites or axons. We'll discuss that in just a minute or two. Actually, we'll discuss it now. The two types of processes are axons and dendrites. Here's the cell body, and these spiked, forked processes are the dendrites. They're like antennae. They receive input from other neurons, and then if enough inputs are received by this neuron, then it sends out an action potential down its axon. The axon hillock is the first part of the axon here, and it goes down to the axon, and this axon may go all the way down to a muscle in the body, or so it could be a long axon or short axon, depending on how far the muscle is away from the central nervous system. And it ends with terminal branches, which it splits up into different branches to innervate different various muscle fibers. This is what makes up the motor unit, as we discussed in the chapter on muscle tissue. So the dendrites there may be hundreds of short, tapered, diffusely branched processes. This is the main receptive input for the neuron. They receive incoming messages that are directed towards the cell body of the neuron. Here's an electron microscope picture of a neuron with a dendrocytes. Now look at the axon. The axon, usually only one axon per nerve cell. Axons can be very long or very short. At the end of the axon are numerous terminal branches. So it forks out like the branches of a tree. If this is a motor neuron, that way it can innervate multiple motor cells, multiple muscle cells. The terminal regions of the axon are called axon terminals or terminal boutons. Nerve impulses are generated at the axon hillock. Here's the hillock is the beginning of the axon, and transmits down the entire length of the axon. By the time the action potential or the electrical wave reaches the end of the neuron, neurotransmitters are released into the extracellular space, and they're picked up by other neurons. And that just creates a chain reaction from one neuron to the next to convey information. The myelin that we talked about before is produced by the oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system. The function of myelin is twofold. It protects and electrically insulates axons, and it increases the speed of nerve impulse transmission. Here's an example of what the myelin, how it wraps around the neuron. Kind of wraps around it like a Swiss cake roll. Just wraps around and wraps around in successive layers. And what it does, it forms a very tight insulation so that the electrical current that flows down the neuron inside doesn't leak out where there's myelin. It's very similar to the way an insulation of a wire keeps electricity, the electrons flowing within the wire and not leaking out from the wire. In the peripheral nervous system, the plasma membranes of the myelination cells have no channels or no carriers, and they're good electrical insulators. They make discrete units of myelin called myelin sheaths. Between the sheaths are nodes that they're called nodes of Ranvier. That's the space in between the individual myelin sheaths. A Schwann cell in the peripheral nervous system can only wrap one segment of the axon, whereas in the central nervous system, an oligodendrocyte can wrap up to 60 axons at one time. If we were to classify neurons on the basis of structure, there's three types of neurons. There's multipolar, bipolar, and unipolar. Multipolar are the types of neurons we've discussed already. There's one axon and many dendrites. A bipolar neuron has two processes. It has an axon and a process that goes out forth into dendrites, but it starts out as one process from the dendrite. You see the example here, one dendrite and one axon. Of course, each of these fork into different branches, but from the cell body, there's only one axon and one dendrite. Unipolar is if from cell body there's only one process that comes out, and it splits into two, and one of these processes is an axon going in one direction, the other is an axon going in a different direction. These are the type of neurons seen with the sensory nervous system. We could classify neurons based on their functional capability. In that case, we have sensory neurons that are afferent neurons that bring information up from the extremities up towards the central nervous system. We have integration interneurons that process that sensory input. These are found within the spinal cord and within the brain, within the central nervous system. And then finally, we have motor output efferent neurons that affect efferent organs, which are muscles or glands. So these are the neurons that cause the muscles to contract and glands to secrete their hormones. Here's a cartoon of us stating the same thing. So functionally, the neurons are sensory neurons, interneurons for integration, or motor neurons. Note that about 99% of the body's neurons are in these interneuron or integration neurons. These are found in the central nervous system. So all the nerves throughout the arms and legs and the face and the extremities are only make about 1% of the neurons of the body compared to the 99% of the neurons that are found within the central nervous system. Let's talk a little bit about how the nerves conduct the signal down its axon. Let's look at the basic properties. This is going to be very similar to what we talked about when we talked about action potentials in a muscle. It's a very similar process. We have ions. Ions are charged particles. Sodium ions, chloride ions. Sodium ions are positive. Chloride ions are negative. And ions of opposite charges are attracted to each other. When opposite charges are separated, there's a potential energy for those charges to get together. This potential energy is what the cell uses to create action potentials. Action potentials are just the wave of energy that flows down the axon to spread a signal or information from one nerve to the next. So here we have the cell like we saw with the muscle. We have high potassium on the inside of the cell, high sodium outside the cell. Low sodium inside the cell, low potassium outside the cell. And just like in the muscle membrane, in the nerve cell membrane does not let ions cross at any point. The cell membrane is designed so that positive and negative charged ions cannot cross over unless there's a channel. A channel has to be there to allow for passageway. These channels are proteins that traverse the entire width of the cell membrane, and they're selected for certain ions. There's sodium channels, there's potassium channels, and they open under certain conditions. Or some stay open all the time, but most of them are open under certain conditions. A channel, just like a channel is a length of water joining two larger bodies of water, like the English Channel joins the Atlantic Ocean to the North Sea. These channels, they join the extracellular fluid towards intracellular fluid. There's different types of channels that we'll discuss here. This is very similar to what we already talked about with the muscle. There's leakage channels that are always open. There's gated channels which open and close based on some circumstance or some aspect of some event. There's ligand-gated channels which open when a neurochemical or a neurotransmitter binds to the receptor and therefore open it up. This illustrates the ligand-gated. They're closed in this point. The ligand binds to the receptor and it opens the receptor up and lets the potassium ions, sodium ions flow up or down in and out of the cell. There's also voltage-gated ions. Here in this case, this is a closed voltage-gated ion. The membrane is polarized. It has negative charge on the inside, positive charge on the outside. When circumstances dictate that polarization reverses so that they have positive charge on the inside and negative charge on the outside, this is being a voltage-gated channel. It's going to open up due to that change in polarization. That's called depolarization, when the charges swap. And the cell membrane around the ion is depolarized, and therefore the ion opens up, and that allows sodium to rush into the cell. That positive charge rushing in the cell causes depolarization of the membrane next to the ion channel, which then is going to stimulate the next ion channel to open up. So it's kind of a cascade reaction, like dominoes. One affects the others. You look at this slide, but it basically illustrates the same thing that we talked about. It looks at the difference in a chemical-gated ion channel, which is the same thing as a ligand-gated ion channel, versus a voltage-gated ion channel. When the channels open, the ions flow down their concentration gradient and across their chemical gradient. If you have a high concentration of sodium ions outside the cell and a low concentration inside the cell, when that channel opens, you'll have sodium rushing into the cell, bringing all those positive charges inside the cell, and that's what depolarizes the cell membrane with all the positive charges, making it go from negative to positive on the inside, which then stimulates the voltage-gated ion channels next to that to open up and create the cascade from one to the next. There's also an electrical gradient. If you have positive charge on the outside and negative charge on the inside, the positive charge is going to be attracted to the negative charge inside the cell and rush in for that reason as well. Look at the resting membrane potential. This is the membrane potential of a neuron at rest. It's approximately negative millivolts. That means the inside of the cell is minus 70 millivolts relative to the outside of the cell. The membrane is considered to be polarized. It's negative charge on the inside of the cell and positive charge on the outside of the cell. This will be polarized. And that's the resting state. The resting state is made due to the fact that there's different concentrations of ions inside and outside the cell. We discussed that the potassium is concentrated inside the cell higher than outside the cell, but the sodium is a high concentration outside the cell than inside the cell. So this difference in concentration between inside and outside the cell is very important to maintain the resting membrane potential. And another thing is that potassium, I said that there's some leak channels that are always open. Well, there's more of those leak channels for potassium. So more potassium leaks out than sodium leaks in. So if more potassium is leaking out than sodium leaking in, that means there's a net charge of positive ions leaking out of cell. That's what creates the negative charge on the inside of the cell, because more potassium ions are leaking out than sodium leaking in. And there's a potassium pump that transfers 2 potassium inside the cell in exchange for transferring 3 sodiums outside the cell. Again, this helps to replace the potassium that's lost by the leakage channels, and also helps create the negative environment, negative charge inside the cell compared to the positive charge outside the cell. Here's a diagram that may illustrate, there's animation that may illustrate that a little bit better.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
An action potential is the way a neuron sends a signal down the long axon. Action potentials occur in muscle cells as well, as we saw. But look at action potential as an energy wave that starts at the cell body of the neuron and goes all the way down to the end of the axon. An action potential is generated. There's several steps. First of all, we'll look at the resting state. That's the resting membrane potential, the resting state. All the gated sodium channels and sodium and potassium channels are closed, so there's no opening of the channels. We have sodium on the outside with high concentration, potassium inside the cell. The gates are closed, there's no movement of ions. But then depolarization occurs. The sodium channels open up. The sodium channels open up and allow sodium to rush in to the cell. Here's sodium's rushing in the cell, and all these positive charges from the sodium ions create a positive charge on the inside of the cell. Whereas if before it was negative on the inside cell, now it's positive on the inside cell, and that's depolarization. Potassium channels start to open up about this time and allow some of the positive charge inside the cell to escape as the potassium escapes from inside the cell to outside the cell. Then repolarization is when the sodium channels are now inactivated. They've been open long enough so they inactivate. The sodium channels are still open, so that allows sodium to keep going out of the cell. This probably allows potassium to go out of the cell while the sodium is no longer coming into the cell. So that restores the polarization status such that you have negative charge on the inside of the cell and positive charge on the outside of the cell. So it becomes repolarized. In other words, the polarization state is returned. Hyperpolarization happens because some of the potassium channels remain open while the sodium channels reset. So the potassium channels stay open longer than the sodium channels, and that allows for more potassium to leak out, and therefore it gets hyperpolarized before it comes back to the normal resting state potential. For an axon to fire, or for an axon potential to start, the threshold of the cell membrane must, or the polarization of the cell membrane must reach a threshold point. Threshold is the transmembrane voltage that's large enough to trigger the action potential. Once an action potential is generated, it's all or nothing. There's no partial action potential. Once it's triggered, it goes all the way from the axon hillock all the way down the axon to the end of the axon. It's all or nothing. Here's an animation that shows the generation of action potential information.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body, in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only, and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
This cartoon shows how the action potential travels down the axon. Here, this portion of the membrane is with resting potential. The membrane is polarized. It has negative charges inside, positive charges outside. But let's say that these channels have opened up from some source, maybe a ligand-gated channel has opened up, and this might be at the neuromuscular junction or the synaptic junction. But it's such that the sodium rushes in, and now the positive charges are located inside the cell, and negative charges on the outside the cell. So now this membrane has become depolarized. This depolarization triggers, along the membrane here, there's other voltage-gated cells that are nearby that they sense this depolarization, so then they open up. Just like this, they open up and they allow sodium to rush into the cell in the area next to the other channel. And that stimulates the next channels down the next channels to open up because they sense the nearby depolarization as well. So it just cascades from one segment of the axon to the next segment of the axon. That way, I compare it to dominoes falling, how one segment creates the action of the next segment to it. This sort of illustrates the same phenomena, how the membrane is inside the axon is negative, positive on the outside. At one point, the sodium channels open up, sodium rushes in, and then the polarity reverses, so now you have depolarization, positive charge inside, negative outside, and then that depolarization triggers the calcium channel. Triggers the sodium channel. The next sodium channel to open up, and repeats the process, so that now you have more positive charges inside the axon at this point of the axon, and it keeps on going, so on and so forth, so that it just propagates down the axon. One ion channel at a time opens up and allows for sodium to rush in, which stimulates the next ion channel to open up. Once action potential is started, there's no stopping it. It conducts all the way down the entire axon length. It's basically saying the same thing. Each successive segment of the membrane depolarizes and then repolarizes. So look at the segment, one segment at a time, all the way down the axon.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
So now we're talking about refractory period. A refractory period is the time in which the neuron cannot trigger another action potential. It usually comes after an action potential has been triggered. There's an absolute refractory period and a relative refractory period. The absolute refractory period is the time when the sodium channels can't open up again after they've been opened. It takes a period of time for them to reset. So if you stimulate the nerve and you start an action potential at the 1 point nerve, if you re-stimulate the same nerve, it's not going to be able to fire again for a period of time until it gets into the relative refractory period. The relative refractory period follows that absolute refractory period. By this time, some of the sodium channels have returned to their resting state, so now they're able to be opened up to allow for influx of sodium. So that, in this case, the action potential can be reinitiated. There's two factors that affect the speed of action potential propagation down the axon. One of those is the axon diameter. The larger the diameter of the fiber is, the less resistant it is to local current, therefore the faster the speed of conduction. And the degree of myelination. The more myelinated a neuron is, the faster that nerve conducts electricity. So those two factors, axon diameter and degree of myelination, control the speed of conduction of nerves impulses down the axon. This was kind of covered in that last little video, but the myelin sheaths prevent leakage across most of the axon membrane, and between the axon, between the myelin sheaths, there's these nodes of Ranvier, and in these nodes there's a lot of voltage-gated sodium channels located at these areas. So these are just waiting to open up to let the sodium inside, and so the action potential seems to skip from one node to the next node because it's not able to leak out through these myelin sheaths because that's such a good insulator. So it's like it bounces from one node to the next, and that increases the speed significantly. Like the animation showed, it can go up to 100 meters per second with the myelination compared to 1 or 2 meters per second without myelination. This just shows that how in the nodes of Pranvé that's where the sodium input goes in from outside the cell to inside the cell. Sodium ions are these represented by these circular spherical structures that bring their charge inside, and they can't leak out where there's myelins, so they're able to propagate down faster and go to the next node. And then the next node gets triggered by the depolarization, so the sodium channels open up there and can have a cascade of the action potential from one node, bouncing from one node to the next node. Instead of going continuously down the axon at all segments, it skips from one node to the next node. That's what makes it go faster. The synapse is at the end of the neuron. The neuron has to connect to other neurons to pass information on to other neurons. This is accomplished by a chemical synapse. Here you have a picture of a nerve with its axon terminating on another nerve. So this will be the synapse. The synapse is basically connection between one nerve and another nerve. Terminology: a presynaptic neuron and a postsynaptic neuron. Presynaptic neuron is the neuron that sends information to the synapse of the other neuron. The postsynaptic neuron is the nerve on the receiving end of the synapse. Note that if there's a nerve, this nerve is connected to another nerve, this nerve could be the postsynaptic neuron for one synapse and the presynaptic neuron for another synapse. So presynaptic neuron just refers to the nerve that triggers the other second nerve to start its action potential. Postsynaptic neuron is the one that receives the information from the previous nerve.
There is a narrow gap, the synaptic cleft, between the synaptic terminal of a sending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
Here is a slide that should look familiar. This is almost identical to the slide of the neuromuscular junction that we talked about on the muscle tissue lecture. So this is the presynaptic neuron. This is the postsynaptic neuron here. So the presynaptic neuron, this is an axon of a presynaptic neuron. An action potential travels down this neuron and arrives at the axon terminal. Second thing that happens is it opens up voltage-gated calcium channels that lets calcium rush into the terminal portion of the axon. This calcium influx causes the synaptic vessels to fuse to the external membrane and therefore release its neurochemical into the synaptic space or synaptic cleft, which is this area here. The cleft is the area between the presynaptic neuron and the postsynaptic neuron. The neurotransmitter diffuses across the synaptic cleft and then it binds the receptors on the postsynaptic membrane, just exactly like it does on the neuromuscular junction. Once the neurotransmitter binds to the receptor, it opens up the receptor, allowing for movement of ions in and out of the cell. This results in depolarization of that portion of the cell membrane. Now, the neurotransmitter doesn't stay there forever. It gets reuptake through transport proteins. There may be an enzyme that degrades the neurotransmitter, like we saw with acetylcholine, and acetylcholine esterase inhibits that and breaks down acetylcholine, or it may just simply diffuse away from the synapse. In either event, each of those would result in the neurotransmitter taken away from the receptor, and therefore the receptor would close again. That summarizes all the steps in one slide. Neurotransmitters are the chemicals released from one neuron to stimulate or inhibit the other neuron. There's many different neurotransmitters that have been identified. Acetylcholine was one of the first ones we understood. This is what's released at the neuromuscular junction. It's also used by many neurons themselves. It's degraded by the enzyme acetylcholine esterase. Other neurotransmitters would be dopamine, norepinephrine, epinephrine, serotonin, histamine. All these are different neurochemicals in the brain, and often imbalances of the neurochemicals can result in conditions like depression, Parkinson's disease, or other neurological or psychiatric disorders.